The most incredible aspect of being a scientist is the unforgettable moment where you are the first person to observe a new phenomenon on our planet. It's the unparalleled exhilaration of innovation and thrill of discovery. It's the moment where you realize that your lifelong efforts have resulted in a better life for one person. And immunologists from around the world who have been performing cutting-edge research into one of the human body's most intricate and yet most powerful systems have been at the forefront of this modern revolution in medical science. From the first demonstration of vaccination by Edward Jenner in 1796, to the development of antibody formation theory by Paul Ehrlich in 1900 and the discovery of dendritic cells by Ralph Steinman in 1973, it is clear that the field of immunology has certainly progressed a long way from its early beginnings. However, there is still so much about the immune system that remains unknown, a vast amount of knowledge still waiting to be discovered. Although the fundamental way in which the immune system recognizes pathogens, biological agents which cause disease, is largely understood, the molecular pathways of intracellular signaling in immune cells have not yet been elucidated. Rather, many theories have been proposed that attempt to explain these processes. The current most widely accepted theory for one type of cell, T lymphocytes, is the kinetic-segregation model of T-cell activation. Before discussing the kinetic-segregation model in more detail, it's important to understand the biological context of the immune system. So let's talk briefly about two important immunological cell types before visiting the laboratory of Simon J. Davis and Anton van der Meer at the University of Oxford, where this model of T-cell activation was proposed. Two of the major cell types in the immune system are antigen-presenting cells, or APCs, and lymphocytes. Antigen-presenting cells include dendritic cells and are responsible for internalizing antigen, a substance which induces an immune response, and presenting fragments of it on its cell surface using a protein called major histocompatibility complex, or MHC. MHC antigen complexes are then recognized by a receptor called the T-cell receptor, or TCR, on the surface of T cells. T cells both coordinate the immune response and seek out and destroy the cells that display the recognized antigen. And with that knowledge, let's fly over to the UK to explore the kinetic segregation model itself. Although scientists know the mechanism of T-cell receptor binding to the MHC antigen complex on antigen-presenting cells, how this initial engagement triggers intracellular signaling is not universally agreed upon. This is the focus of the kinetic segregation model, proposed in 1995 by two scientists at the University of Oxford. It is widely appreciated that cells respond to external cues, such as cell-cell interactions and mechanical stresses, by structural changes in the cytoplasmic domains of cell surface molecules. The external cues are referred to as receptor triggering, while the structural changes are considered to occur downstream of the T-cell receptor. At the time of TCR isolation and characterization, most receptor triggering was understood in the context of conformational changes and the formation of receptor dimers, and thus it was broadly anticipated that this structural rearrangement would be experimentally confirmed as the mode of intracellular signaling in lymphocytes. However, Alan Frederick Williams, a noted Australian immunologist, was not convinced. A.F. Williams, in fact, was the first person to emphasize the many aspects of T-cell biology that prevented this model of T-cell activation from being possible. Conformational change-based mechanisms of cell signaling were not exactly his go-to explanations for biological phenomena, his reasoning being because they are invoked to explain everything. He pointed out that the insufficient quantity of major histocompatibility complex on the surface of antigen-presenting cells would actually make MHC TCR binding kinetics so fast that adjacent T-cell receptors could never be co-engaged by two ligands. This provided evidence that TCR dimers would not be formed and therefore proved that this was not the signaling mechanism. Then what was actually happening? Davis and van der Meer argued that it was infinitely more likely that T-cell receptors actually respond to triggering at the level of individual complexes. There were several different discoveries leading to the development of the kinetic segregation model. Firstly, that intracellular signaling is achieved by chemical modifications on cellular proteins. This modification is usually phosphorylation, or the addition of a phosphate group, PO43-, to specific residues or amino acids of the protein. Phosphorylation is mediated by enzymes called kinases, while dephosphorylation, the removal of phosphate groups, is facilitated by enzymes called phosphatases. Another discovery that led to the proposal of the kinetic segregation model was the composition and architecture of the cell surface itself. It was noted that when TCR-MHC interactions do occur, they would need to be segregated from the carbohydrate-rich cell surface barrier, or glycocalyx, because the complex is so much smaller than the components of the glycocalyx. Think of it as a game of capture the flag. The T-cell receptor is one player who needs to find the MHC antigen complex, who is acting as the flag. In order to win the game, the player needs to grab the flag and bring it back to their own base. But players on the other team, acting as the glycocalyx, are forming a huge barrier separating the player from the flag, so there needs to be a way to bring the TCR and the MHC antigen complex closer together. Now envision another pair of teammates, collectively around the same size as the first player and the flag together, acting as a diversion and separating the components of the barrier that prevents contact. This represents another protein called CD2, which forms complexes around the same dimensions as the TCR does, around 15 nanometers in length, and thus separates the membrane into smaller components. This helps to enhance the ability of the TCR to locate and interact with the MHC by forming regions of close contact at the interface of the two cells. Why is this important? Well, scientists needed to discover how the phosphorylation, which causes the signaling cascade, is initiated, and an unexpected discovery confused things further, that tyrosine residues on the T-cell receptor are constitutively phosphorylated. In fact, there was already so much phosphorylation that it was unclear how ligand binding to the TCR could further increase kinase activity to a level sufficient to have any effect. This raised the possibility that it was actually the removal of phosphatases at the time of TCR-MHC interaction that increased the half-life of phosphorylated species in the region, and was therefore responsible for the higher level of TCR phosphorylation upon antigen recognition. So what do all of these ideas have to do with one another? It turns out that CD45, which is a major component of the glycocalyx, is a phosphatase responsible for over 90% of tyrosine dephosphorylation on cell surface receptors. But it's also very large physically, so it would undergo size-dependent exclusion from the close contact regions upon membrane separation by CD2. Therefore, phosphorylated TCR in the close contact region would remain phosphorylated, unlike its normal state, where it is just continuously dephosphorylated by CD45. But one last issue remained. Wouldn't all TCR in this close contact region remain phosphorylated when two cells interacted with each other, thus triggering signaling even if the TCR failed to actually recognize an antigen? It was hypothesized that phosphorylated but non-engaged T-cell receptors would simply diffuse out of the close contact region, therefore allowing rapid dephosphorylation before they can inappropriately initiate downstream signaling.
It is also possible to look at the kinetic segregation model as a series of 5 steps. First, in a resting T-cell, all surface proteins, including T-cell receptors, are transiently phosphorylated by kinases and then almost immediately dephosphorylated by CD45. Then, when the T-cell encounters an APC, proteins at the cell-cell interface segregate in the close contact zone. However, CD45, the chief phosphatase enzyme, is size-dependently excluded. This is where the capture the flag analogy comes into play. Thirdly, the net phosphorylation of TCR tyrosine residues in the close contact zone increases due to the presence of kinases but the absence of phosphatases. Fourth, the separation of the cellular membrane by CD2, which in the analogy would form a diversion to allow contact between the player and the flag, enhances the scanning ability of T-cells. TCR binding to the MHC antigen complex prevents its diffusion out of the close contact zone, while unproductively engaged but phosphorylated TCR diffuses out of the region and is dephosphorylated, preventing the inappropriate triggering of an immune response. And finally, the longer half-life of engaged and tyrosine phosphorylated T-cell receptors allows for the triggering of the signaling cascade. This process involves the recruitment of another tyrosine kinase, ZAP70, which in turn leads to the further involvement of other kinases and the phosphorylation of other intracellular signaling molecules. This entire process culminates in T-cell activation and the immune response. That is the essence of the kinetic segregation theory.
Science always moves forward, and one theory is often replaced by another as new evidence is uncovered. But at least one of immunology's greatest riddles now has an explanation, thanks to the groundbreaking research of Drs. Simon J. Davis and Anton van der Meer at the University of Oxford. It is part of the very nature of science that one day, perhaps dozens of labs, hundreds of experiments, and thousands of observations later, we will have an even better understanding of the mechanisms of T-cell signaling. Who knows? It could very well be me who writes that paper that adds to our knowledge of immunology, and it could even be you. However, one thing's still for sure. If our goal is to solve all of immunology's mysteries, then the kinetic segregation model was a huge leap in the right direction.